Doing something a little bit different today. I'm a senior bio major at my college, and so this was my senior presentation project. And my grandparents wanted to come see it, but they weren't able to, so I told them I'd record myself giving the presentation and then get it to them that way. Because email does not like files as big as this one's going to be, I thought it could be kind of fun to post it to YouTube. Because I've been wanting to do some videos where I talk about my special interests anyway, so this seemed like a good way to start doing that because I had the presentation ready made. So yeah, without further ado, here's my senior presentation. Resurrection biology, what is it and should it be attempted? Oh, also I'm not using my camera because this presentation wasn't originally made for YouTube, and I didn't want to have to go back through and rearrange everything just for the purpose of using my camera, so just audio today. Alright, first up, what is resurrection biology? It is an attempt to revive an extinct species. To do this, you need to recover genetic material, which is difficult in and of itself. DNA doesn't really fossilize, and it does have a half-life, so the older something is, the harder it's going to be to extract that DNA. And even if you do recover any of it, also I've got a bit of a cold, so sorry if I sound weird. If you do recover any DNA from an extinct animal, it's quite likely fragmentary, so you're not going to get like the full genome or anything. To get around that, you fill in the blanks with close living relatives. After you've done all that, you clone it, and then you gestate whatever cloned embryo you're able to produce, using again a close living relative as a surrogate. And then what? Is the embryo viable? Does it develop to full term? Can it survive outside of the womb in this case? Most of these cases today are mammals. Do you make more of them? Can they reproduce with each other? Can the species ever sustain itself without human intervention? Can you return it to its former habitat? These are all questions that need to be considered and addressed before any attempt can be made to revive a species, and we'll go into some of those today. Functional de-extinction is the revival of an extinct species or producing a sufficiently close proxy which is capable of successfully resuming the extinct species' former niche. What this means is we cannot simply make an organism that looks like the extinct animal. We have to produce an organism which can actually fill the extinct species' former place in the environment. Something that can live in the same sort of habitat, go after the same sort of food source, and be a food source for the same sort of animals depending on where it was at in the food chain. Somatic cell nuclear transfer is the method by which multicellular organisms are cloned. The basic steps are removing a somatic cell nucleus, and a somatic cell is any body cell that isn't a sperm or an egg cell, so any non-gamete cell. You remove the nucleus from a somatic cell of whichever individual you're attempting to clone, and you also remove the nucleus from an oocyte, an egg cell. You transfer the nucleus of the somatic cell into the now empty oocyte, induce that oocyte or egg cell to begin dividing, and you implant it into a surrogate where hopefully it will develop into an organism. Now, with living species, this process is not easy necessarily, but fairly straightforward and has been done multiple times, like in the case of Dolly the sheep. But if you're attempting to do this with an extinct species, it gets a little bit more complicated because, as I mentioned, you will rarely find a whole intact nucleus in an extinct animal. So you have to settle for whatever DNA you can recover. And then from there, of course, you'd be filling in, editing the genome with to include the traits of a living relative in order to fill in those blanks. And then you would also have to take the oocyte from the close living relative because you wouldn't have any egg cells on hand from the extinct species. And of course, the surrogate also has to be from a close living relative because part of the problem is that there are no living individuals of the species left. So it gets quite a bit more complicated. The way DNA is edited in these cases is with the CRISPR-Cas9 system. It's a precise gene editing tool wherein something called a guide RNA is synthesized, which is a single-stranded RNA sequence which is complementary to, combined with, whatever sequence of DNA you want to edit out. The guide RNA is used to program the Cas9 protein complex, which can then travel along the DNA until it finds the section which matches its RNA guide, and then cuts out that section very precisely. From there, the DNA, of course, wants to repair itself, which it can do either by sort of rebinding the either side of the break point, so it just sort of gets smaller, or a new sequence can be added. So to edit genomes, what scientists will do is pick which gene they want the Cas9 to cut out and insert a different sequence of the gene. So in this case, it would be perhaps adding genes from an extinct animal into the genome of one of its living relatives, or even vice versa, depending on how complete the recovered genetic material is. And that will give whatever organism is produced traits from this extinct species, perhaps to the point that it can functionally resurrect the species. One recent attempt to resurrect an extinct species, which got a lot of press, is Colossal Bioscience's purported resurrection of the dire wolf. Now, the dire wolf, Enochion dirus, was a large muscular canid species endemic to the Americas, so only found in North and South America. It went extinct around 10,000 years ago, towards the end of the Ice Age, along with quite a few other things. It had a similar morphology to living gray wolves, so it looked very similar, but it was not actually a close relative. It was a canid, a member of the dog family, but it was no more closely related to gray wolves than to foxes or jackals or, any other member of that family. And actually, if you look, I've noted that it used to be known as Canis dirus. That is important. Canis is the genus name of the genus to which modern wolves belong, dingoes, domestic dogs, coyotes, and it signifies a more closely related group of animals. The dire wolf used to be classified as a member of this genus, but now is in its own Enochion, which is significant because that means it is not considered a close relative of gray wolves and dogs and their relatives. It had a similar skeleton morphology to modern gray wolves, though it was more robust, especially with its skull, which was broader. So it's believed it might not have been as fast, but it was probably more muscularly powerful than a modern gray wolf. In 2025, the biotech company Colossal Biosciences announced that they had created 3 new dire wolf pups. 2 males, Romulus and Remus, and a female named Khaleesi. The pups were produced using somatic cell nuclear transfer, which I mentioned earlier, with gray wolf DNA edited using CRISPR to produce more dire wolf-like traits, so increased body size, and they modified the pups to have white coats, though an independent study did note that the most likely coat color for dire wolves, given their habitat, was actually most likely a reddish brown, so the white coat seemed to have been more of an aesthetic choice, but I can't confirm if that is true, if it was truly just an aesthetic choice. That same study confirmed that the pups are in fact gray wolves, Canis lupus, with less than 0.1% of their genome edited to produce the changes that I described. So they are not true dire wolves, they're just larger than average gray wolves. One of the attempts which resulted in a true clone of an extinct species is the species that went extinct twice, the Pyrenean ibex. This subspecies of the Iberian ibex went extinct in the year 2000, but DNA was recovered from the last known individual before she died. Using this genetic material, a fetus was successfully cloned in 2003. It was one of nearly 300 attempts. This is the only one that was successfully carried to term. However, the ibex had faulty lungs, leaving the and died within mere minutes, giving the Pyrenean ibex the tragic legacy of the only species that has ever gone extinct twice. One of the most famous faces of the resurrection biology conversation is the thylacine, Thylacinus cynocephalus, also known as the Tasmanian wolf or Tasmanian tiger. Wolf for its superfluous resemblance to placental gray wolves, and tiger because of the stripes along its back. It was the largest carnivorous marsupial of the Holocene until an extensive bounty program by the British government of Tasmania led to the species extinction in 1936, when the last individual died in a zoo. The species was formally declared extinct 50 years later in 1986. The reasoning behind the extermination program was because the thylacines were believed to be a threat to British livestock, although it has since been determined that they likely did not possess the jaw strength necessary to actually eat a sheep, and the most likely candidate for any livestock depredation is in fact the dogs the British brought with them themselves. In the year 2022, a hybrid genome for the thylacine was published. Because they've gone extinct so recently, there are plenty of preserved specimens in museums from which DNA can be extracted. Gaps were filled in using genes from the Tasmanian devil, which is actually the current largest marsupial carnivore. Now, would the Tasmanian devil be a viable surrogate for any thylacines produced? Well, thylacines were, on average, over twice as large as the Tasmanian devil. And despite the fact that marsupial babies are born quite small and then gestate for the rest of the time in a pouch, without knowing the relative sizes of thylacine and marsupial joeys, thylacine and Tasmanian devil joeys, once fully developed, it remains to be seen whether a Tasmanian devil could be a viable surrogate for any developing thylacine if produced. Now, perhaps the most famous face in the resurrection biology conversation is the woolly mammoth. This relative of the elephants had a range extending across Eurasia and North America. It went extinct, again, around the end of the Pleistocene, the last ice age, about 10,000 years ago. Its closest living relative is the Asian elephant. Like its modern relatives, the woolly mammoth is believed to have been an ecosystem engineer. What this means is a herd of mammoths could have significantly altered their ecosystem simply by their presence. So distributing the seeds of their preferred food plants as they walked and digested them and deposited them once they were done digesting. Knocking over larger vegetation and making room for shorter vegetation, perhaps providing habitats for other animals. So their impact was likely massive in the Pleistocene ecosystems. Despite going extinct 10,000 years ago, we can actually extract quite a bit of genetic material from mammoths because we find them preserved in permafrost. What's important about this versus other kinds of fossils is their soft tissues are preserved, including things like skin, hair. You can see in this picture, their hair was sort of a reddish blonde color. Stomach contents is preserved, so we know quite a bit about their diet. They preferred things like grasses, lichens, certain shrubs. And we can extract nuclei. In 2019, researchers in Japan injected mouse oocytes with nuclei recovered from frozen mammoths, and the oocytes actually started to divide. Now, they didn't divide all that many times, but the fact that they started to at all means that if better preservation for the nuclei can be, it provides some groundwork for increased research into the possibility of producing successful mammoth clones from such recovered DNA. If these nuclei were once transferred into an oocyte from a mouse, divide. Of course, they would not be mouse oocytes if it were an actual cloning attempt. It would be from Asian elephants, their closest living relative. But the fact that these cells could divide once injected with mammoth nuclei is huge, for potential resurrection of the species. However, before any attempt to resurrect an extinct species, there are multiple ecological concerns that must be addressed. The question of does its habitat still exist is perhaps best exemplified by the woolly mammoth. The world has warmed and the glaciers have retreated since the Pleistocene when the mammoth went extinct. And the northern parts of the world are warming faster than the rest of the globe. Does any facsimile to the Pleistocene ecosystem exist in the modern world? Well, a study done in 2024 determined that this region of the North Slope of Alaska may be one of the closest environments we have to the mammoth steppe of the Pleistocene. However, it determined based on available plant matter that this area could potentially support a population of several thousand mammoths, up to about 40,000. However, the researchers themselves note that this estimate was based purely on available vegetation and did not take into account competition or predation, all of which are important considerations when talking about a species' carrying capacity, how many of them can actually survive in an area. It's introducing an invasive species into an environment tends to have disastrous consequences for any local wildlife. In this case, we would be introducing multiple multi-ton herbivores into an environment which already is facing high pressures due to climate change and melting ice. If the mammoth, and we know mammoths were social, so it would have to be a herd that was introduced. They couldn't simply survive on their own. So if they are better at getting the vegetation they need than the native caribou and musk oxen, then with their higher energy demands as well, it is likely they could potentially outcompete caribou and musk oxen for the vegetation in that area, causing harm to the species that are already there. In addition, the mammoth's natural predators are also extinct, the smilodon and the dire wolf. The predators in this area now are Arctic wolves and occasionally polar bears, depending on how far south they go. The wolves already go after caribou and musk oxen. It is possible they would not even recognize mammoths as prey, and even if they did, it's highly likely caribou are an easier target than mammoths are. And so if the mammoths are outcompeting the caribou for food and then are not facing the same predation pressure from Arctic wolves that caribou face, then the mammoths could outcompete the caribou and perhaps even the musk oxen. On the flip side, it's possible it could be outcompeted by native species. We don't know how close this area actually is to the mammoth's environment in the Pleistocene. This is just our best guess as to what might be close. If we're wrong, then they might not know how to survive. They might not be able to gather the food they need. And if the native predators do recognize them as prey and they can't find food for themselves, then they may simply follow the Pyrenean ibex and go extinct twice. In addition, we know they were likely social and highly mobile. We don't know how big the herds were, and we don't know how far they traveled. So, how much space do they need? How many do we need to make in order to make a decent-sized mammoth herd? This is still conjecture. We can use modern elephant herds as an approximation, but we don't know how close of an approximation that is. So, additionally, important for functional de-extinction is, can they reproduce? The direwolf pups produced by Colossal Biosciences were birthed by cesarean section. This was apparently planned likely because large offspring can lead to birth complications. They were birthed from dog surrogates. If the direwolf traits are to be spread amongst a population of wolves, the pups will have to reproduce eventually. And it's possible if the large genes do pass on, then normal wolves or dogs or whatever they end up reproducing with will not have a sufficiently sized birth canal, leading to birth complications and possible death of offspring and mother, making it so the species these traits cannot be perpetuated in a population. Additionally, as I mentioned, these extinct genomes usually have to have gaps filled in with a close relative. Well, hybrids are often infertile. So depending on how much of a hybrid it is, how much DNA has to be replaced, and how close the living relative is, it's possible whatever clone we produce cannot itself reproduce. It may not be able to produce offspring, which means the species cannot self-perpetuate and therefore will not survive without human intervention and cannot resume its former place in its environment. Therefore, again, we have not functionally resurrected the species. We have just created a look-alike. And that would not be meeting the goal. The all-important question, is there sufficient genetic diversity? Tasmania separated from mainland Australia about 10,000 years ago, and while this means the thylacines on Tasmania survived the eventual extinction that would befall their Australian counterparts 3,000 years ago when humans arrived in Australia, it did lead to the Tasmanian thylacines undergoing something of a population bottleneck. They became reproductively isolated with an ever-dwindling gene pool, so that by the time the Tasmanian thylacines finally followed their Australian relatives, there was incredibly low genetic diversity amongst the species. A study conducted on 12 preserved thylacine species found them to be about 99% identical in certain key variable regions, meaning that this effect would only be exacerbated in any thylacines produced through cloning. And this lack of genetic diversity would lead the vulnerable to birth defects to disease and a host of other problems, which could again lead to them simply going extinct all over again without human intervention. The relative used to fill in the gaps in the thylacine genome produced in 2022, the Tasmanian devil, faces a similar lack of genetic diversity now, which has actually left them vulnerable to a contagious facial cancer. It's called devil facial tumor disease, and the cancerous cells themselves can actually pass between individuals. Usually, it's thought to usually occur during fighting or communal feeding. So even if we produce thylacines that have some of their own DNA and some devil DNA, it's possible we could simply be leaving them vulnerable to an additional problem of this facial cancer. So again, if there's insufficient genetic diversity, then the species cannot perpetuate itself. It possibly could never return to its niche because the risk would be too great of its just going extinct again. And again, we have not functionally resurrected the species. We have created a look-alike, which in all likelihood can only survive in a zoo. In addition to ecological questions, there are questions of ethics which are vitally important to consider before attempting to resurrect a species. As mentioned, the one successful clone of the Pyrenean ibex died in minutes, likely in great pain, due to faulty lungs. Guidelines for experiments involving animals include no causing of undue pain. Is it ethical to continue attempting to create clones or facsimiles of extinct species, knowing it is possible we simply doom them to short lives filled with pain because they can't breathe or some other vital function cannot function properly? For mammoths, if their modern relatives are anything to go by, they were likely highly intelligent and highly social. Is it ethical to run the risk that, like in the case of the ibex, we could only produce one and it would live alone? Or run the risk of them only being able to survive in a zoo? Is it ethical to create something, this new species, only for it to never be able to resume where it used to live in artificial conditions, possibly lonely or bored? In the case of the thylacine, is it ethical to attempt to resurrect the species knowing we may just be dooming them to genetic defects caused by low genetic diversity and leaving them vulnerable to disease, which could simply lead to them, going extinct again? Even in the case of their modern relatives, there are ethics to be considered here. Asian elephants and Tasmanian devils are both, themselves endangered, facing population decline. Is it ethical, under these conditions, to remove breeding females and oocytes from populations which are already struggling, on the off chance that we might be able to produce a functionally de-extinct member of their dead relatives? What if elephants are pregnant for 2 years? If we remove enough Asian elephant females to serve as surrogates for a potential herd of mammoths, that's potentially quite a bit of possible breeding time and offspring that could go to helping this endangered population recover. Tasmanian devil, same thing. They're facing a population crisis. They're not pregnant for as long as elephants are, but there's still the possibility, if we remove healthy breeding females and take their egg cells to produce thylacines. Because, again, the ibex was one of nearly 300. Who knows how many it will take to produce a viable individual of these extinct species. So is it ethical to risk worsening the already pretty bad conditions of the populations of Tasmanian devils and Asian elephants for an off chance at resurrecting their extinct relatives? Is it ethical to use our time and resources that could be put to work developing new strategies for conserving species which have not yet gone the way of their extinct relatives, and making sure they never do, making sure the Asian elephant never goes the way of the mammoth, making sure the Tasmanian devil doesn't go the way of the thylacine? Is it better to learn from our past mistakes and ensure that they aren't repeated, rather than pursuing the correction of mistakes that have already been done, risking the worsening of damage, which is not yet irreversible, in an effort to correct damage which has already been done and will likely never be reversed? I would say no. I would say it's better to devote our time and our resources to preserving what is still here, rather than bringing back what is gone to soothe the guilty conscience or satisfy, even though it could be amazing to see these species alive again. They're gone, and in all likelihood, they will never be back in a capacity to resume their former niche, and therefore likely should not be brought back at all. It doesn't seem ethical to produce an extinct species again, only for it to be unable to survive outside of a zoo. With that, here are the sources I used for this, in case anyone would like to look at this further. It's a fascinating topic. Kind of ironic. This video is actually at the time limit when I gave this presentation in real life. It was a little bit short on time, so that's funny. There's things I wish I would have added to the presentation when I gave it, things about legislation and regulation and whatnot, and I thought about going through and adding those before I posted this to YouTube, but I decided you guys should get it in as close to its original form as possible. I think I phrased some things differently, but you get the presentation as it originally was. If you're still here, thank you for watching this whole thing, and yeah, hope you like it. Bye.